So today we begin talking about organic chemistry, which we're learning about carbohydrates, lipids, nucleic acids, and proteins. There is an entire course at the school offered in organic chemistry. This is nowhere near the level of complexity that that course offers. This is sort of organic chemistry for dummies. Not saying you're dummies. I'm just saying I've really, really lowered the bar here so you have a basic knowledge of organic chemistry in order to survive introductory biology. So organic chemistry is the science of studying carbon-containing molecules. That's it. Molecules that contain carbon. Typically, they'll have these cool rings to them. It's called organic because it was based off of this idea of vitalism. That is, there are certain molecules in the body that can only be created by something that has a vital life force, a vital life essence, a soul. This is not accepted anymore, just so you know, but this is sort of a historical grounding in where this came from. Organic is not based off of, and this is a common mistake recently, the idea of organic foods. Totally different things. Organic foods have their own designations. Organic chemistry is studying molecules that have been traditionally associated with needing a life force or with growing within the body. There's this awesome scientist by the name of Friedrich Wohler. He tried to create ammonium cyanate, which is NH4OCN. I know, you don't have to know that, but I'm just telling you. He had studied this other molecule called urea forever. Now, if you ever go into graduate school, if you're a biologist in graduate school, your role in life becomes to do whatever your professor says. If they say jump through any hoop in front of you, you jump through that hoop. And what Wohler had to do was he had to study urea crystals. Urea is sort of the same crystal that forms from, think, urine. Does that make sense? So he's looking at pea crystals under a microscope, day after day after day, year after year. And then, like most biologists, when he got, or most scientists, when he got out of graduate school, he swore never to work with it ever again. With my graduate school experience, I dissected 46,320 fruit flies. And I had to count their eggs. Now, you guys have seen dissections before. Anybody done a dissection in here before? A few of you guys. Frog, something like that. Something, the worm maybe. A fruit fly is really, really small. They live around your bananas. Now, if you've seen these fruit flies on bananas, they're very large compared to the ones I was studying. I had to use a very, very fine-tipped needle and another needle to dissect them under the scope for 8 hours a day, sometimes 15 hours a day, almost every day of the week. It was terrible. I never wanted to look at a fruit fly again. So, of course, this semester, I decided, well, the best way to teach genetics is with fruit flies. And I started having flashbacks. But anyways, Friedrich Wohler, he looked at this urea. This guy knows the way urea looks. And he must have thought he was having flashbacks, because when he tried to make an ammonium cyanate, he saw urea crystals. At that point, nobody thought you could make urea in the lab. They thought that it was necessary to have a vital life force to create this urea. So this discovery sort of said, now we can make things in the lab we never thought possible. This is what started organic chemistry. So like I said, organic chemistry is the science of studying carbon-containing compounds. And we study carbon-containing compounds because carbon is a very, very versatile atom. Carbon can form 4 covalent bonds. It can bond to lots of different functional units. And carbon atoms can have multiple forms. Carbon-containing molecules can have many, many different forms. So it forms 4 covalent bonds. It can bond to lots of different functional groups. And it has multiple forms. In case you haven't recognized it yet, whenever I put big lists on a board like this, you should know them. Kind of important. Granted, I do say that about literally everything that comes out of my mouth. watching you guys write furiously. Don't worry, we're going to go through each of these individually as well. For instance, carbon bonds has 4 possible bonding points. It has 4 electrons in its outer shell. So if you remember, the carbon is C, 6, and 12. It means it has 6 protons. That means to be electrically neutral, it's also going to have how many electrons? 6 electrons. So you got is 6 protons in the nucleus. Hey, based on this, 6 and 12, how many neutrons are in the nucleus? 6. 5, 6. And then you have 6 electrons. In the first shell, how many electrons fit there? 2 electrons fit in the first shell. In the valence shell, the outer shell, then, you have 4 remaining. 1, 2, 3, 4. Those 4 electrons allow these carbon-containing molecules to connect to each other in many different and wild ways. You can have single bonds forming between carbon. Up here in this corner, we got single bonds. So we got hydrogen, 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 carbon. Single bonds, 4 possible connections to each of that carbon. You might have a double bond up in the corner, that carbon attached to the oxygen. Over here, we've got C2H4. We've got a double bond forming between the carbons. And then down here, we have a triple bond forming between carbons. Which one of those is the weakest? The single bond. Which one of those is, say, the tightest, the one that brings them atoms closest together? The triple bond. So you can have different bonds between carbon atoms. Single bond, double bond, triple bond. Occasionally you might even see a quadruple bond, but that's extremely rare. Because of the way it can bond as single or double bonds or triple bonds, you have carbon molecules with different forms to them. And you have different forms. What else do you have? Different functions. You might have a linear chain. Carbon after carbon after carbon, one after the next. Now here we have carbon and hydrogen attached to each other. What do you know based on the last lecture about carbon and hydrogen's interactions with each other? How do they share electrons? That's a hard one. How about if I put it this way? If carbon and hydrogen share electrons evenly, what must be relatively even between them? Well done. Electronegativity. So if these guys have similar electronegativity, these carbon and hydrogens, is this molecule going to be hydrophilic, hydrophobic, or amphipathic? Welcome to the type of questions that will be on the exam. Now you just got a 1 out of 3 chance, even if you don't know it. Why would it be amphipathic? What does it mean to be amphipathic? You have both a polar side and a non-polar side, right? Are there poles to this? No. So it's not amphipathic. Now we're down to 50-50. Thanks for throwing it out there. Say it again. Hydrophilic. Hydrophilic molecules, what do we know about them? They're polar. Do you see poles? No. Thus, not hydrophilic. So what is it? It's hydrophobic. Well done. Once you've narrowed it down to the only answer left. This is a hydrophobic molecule. It has evenly shared electrons. It has no poles. Thus, it's not going to, the water molecules have nowhere to attach. They have nowhere to bind. You might have these cool rings. See those rings up in the corner? Those are carbon rings. Those are indicative. Those are sort of a huge deal when it comes to organic chemistry. Carbon rings are what most chemists study when they study organic chemistry. And you could be highly, highly branched in this far one. So I guess that whole non-polar, polar thing really would have been better to talk about on this slide where it gives you the answer. But you know. Carbon and hydrogen, because of their similar electronegativities, will evenly share electrons. And if they're evenly sharing electrons, there is no pole. There's no temporary pole. Thus, they are nonpolar. They do not dissolve readily in water. Wow, I wonder if there's supposed to be a picture here. Functional groups are groups of atoms with characteristic chemical and physical properties. Now here's the deal. Functional groups can be attached to a carbon atom. Oops. The way I always think about these, let's see, the best way to think about these. Best way to think about this is, how many of you guys have gone to a wedding? Good, okay, a few of you guys. How many of you guys have been married? Okay, a few of you. When you've gone to these weddings, you may have noticed that there's always the exact same mixer on people's registry. And do you know the mixer I'm talking about? It's like the KitchenAid super awesome $300, $400 mixer. Do you guys know this mixer? Okay, just some of you guys, you may have seen this mixer somewhere, in a store perhaps. But this is a giant mixer. Everybody hopes for it because reasons. And it looks sort of like, got this arm coming off of it, a head, and this little plate down here. That's what the mixer looks like. Okay, sort of. I'm not good at drawing. There's a mixer. This piece here would be like the functional group. Or this would be the carbon. A functional group will attach to it. You can attach lots of things to a mixer. Okay, so if you guys don't know this mixer, you guys have the mixer, the like $15 one from Target, right? Okay, it looks like this. And it's got a little handle there. And then you put stuff right here. What do you put there? A whisk. Sure, you put a whisk on. What's a whisk good for? For those of you who have the expensive one, I'll put it here. Whipping stuff. What do you whip? Going with food here. Cream, eggs, things like that. You can beat them. All right? What else can you put there? You can remove, that's really, it's not terribly good for like mixing dough. You can't make cookies with that. Have you tried? If you did, you destroyed it. You also might have, the ones that look sort of like that. That they spin around, go figure, right? Do you guys know this one? Yeah, you put that on, you spin around, now you can make cookie dough. Now you can make a cake. And you got this weird one that's sort of like this corkscrew screw thing like that. Do you know what that's for? Nobody knows what that's for. Yeah, that's what they say. It's a dough hook. Yeah, nobody knows how to use it. Nobody's ever used it aside from to look at and be like, well, that was a very expensive mixer. I'm going to have to keep this in my drawer for the rest of my life now. I've made a commitment. Each of these represents a different functional group. They can bind to the mixer and give that mixer different properties. If you don't want to think about mixers, I've got this like a weed whacker type thing that does the same thing. I've got this attachment that makes it a weed whacker, an attachment that makes it into a tiller, an attachment that makes it into a hedge trimmer. It's really cool stuff. I only ever get parts for it at Father's Day because apparently they can't think about anything else I would like. And my wife's trying to hint at something about that. Maybe I should go and do something outside. So, functional groups can be attached to a carbon atom to give the molecule certain functions. And you've got a lot of different functional groups. You've got a methyl group, hydroxyl, amino, carboxyl, aldehyde, phosphate, sulfhydryl, and phenyl. The methyl group, so that's the very top. Typically, you're going to see this in alanine. It's an amino acid over here. You've got a hydroxyl group. Just look at the name, hydro and oxyl, right? Like hydroxide. And what's the formula for hydroxide? Remember, we just studied that last time. What? OH. And sure enough, hydroxyl is OH. Moving down, amino, carboxyl. I like carboxyl because it says COOH. C-O-O-H. And aldehyde, which we'll learn about in lab. Phosphate group. How do you know it's a phosphate? It's got a big P in the middle. sulfhydryl. It's got sulfur and hydrogen in a phenyl group. Each of these conveys distinct chemical properties to the molecule that they get attached to. And so, to give you a heads up on the exam, I'm going to pick a few of these, and you are going to have to match the name to the structure. So I'm going to give you a section on matching. I do this all the time on my first exam, and people either get the whole thing as like super easy points, or they miss every single one, and there is no in between. If you want to see bigger pictures of these, go ahead and look, go on Google, it's there, look in your textbook, it's there. Just write in functional groups, and these pop up. You're not going to have to recognize it in a structure, you're just going to have to see it separated, so it's sort of like just looking at this middle part of the screen here. That's all I want you to know. Be familiar with it. So you got these four attachment points where functional groups and other parts of the molecule can bind. And then finally, carbon can have multiple different forms. When you have two structures that have identical molecular formulas but different structures and characteristics, you have what are termed isomers. Now take a look at this one on the far left. How many carbon are there? Not trying to trick you. 4. How many carbon in the middle one? How many are in the last one? 4. 4 carbon in each. Oxygen in the first one. 1, 2, 3, 4, 5, 6. 6 in the first. 1, 2, 3, 4, 5, 6 in the second. 1, 2, 3, 4, 5, 6 in the third. Hydrogen. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Each of them has the exact same molecular formula, but they have different arrangements. They are slightly different from each other. This bottom one, these two in the middle are mirror images of each other. Here, though, you have both arms facing in the same direction. So isomers may have the same chemical formula, but they are in fact different forms. And if you have a different form, what do you have? Different function. One example of this was, what's it called? They called them the flipper babies. It was, I can't believe this is just slipping my mind. Thalidomide, that's what it was. So thalidomide was a medication for nausea during pregnancy. Now, this was initiated over in England. In England, before you're able to give any sort of medicine to people, you need to give it to lower animals first, mice, rats, rabbits, things like that. And they tested on mice, rats, and rabbits, and mice, rats, and rabbits were able to produce totally normal kids. They started to give this thalidomide out to people and said, great, this will reduce your nausea during pregnancy. They tried to market it in the US. Well, the US has a much stricter drug enforcement policy. So when it got here, they said, well, I see your data on mice, rats, and rabbits, but where's your data on dogs and primates? And they said, we don't have that. We're from England. We don't worry about testing those things. And they said, well, you have to test them now. So in order to sell in the US, they started testing on primates and dogs. And what did they find was that thalidomide caused serious birth defects in primates and in dogs. So in the US, we said, okay, we're not selling that to people because it's going to mess up their kids. In the UK, there was an entire generation, a few years born, where they'd subscribed to thalidomide for nausea, and those children turned out with serious birth defects. They didn't have arms. Their hands grew out of their shoulders. Their feet grew out about 3 years. 3 years. Not like a 20-year generation. Like, they didn't go on to reproduce after that. They could have. I don't know. There might be thalidomide babies out there. But for a few years, there were a whole bunch of kids born with serious birth defects, to the point where they did call them all the flipper babies, which is really mean to do. We have two kinds of isomers that I'm interested in you knowing: structural isomers and stereoisomers. Structural isomers have entirely different structures. They have different bonding arrangements. So you can see you've got carbon here is straight linear here. The carbon's linear except for that one going off the top. It is a very, very different form. The way I think about structural isomers is I tend to think about Legos. You guys played with Legos before, right? If you were to take some Legos, you can take, it's falling apart. You take Legos, you build some structure. Say you build a car. Now that car has certain function to it, right? It rolls, it drives places, life is good. If you were to remove the wheels and put them somewhere else, rearrange all of the Lego pieces, you could build a jet out of it. Jet has different function than a car. Jet flies. You've pulled it apart and rearranged it. All those original bonds have broken. You've had to break things in order to put it back together in a way that's meaningfully different. That would be a structural isomer. Two different, you're using all of the same components, but putting them together in an entirely different way. The other type of isomer is called a stereoisomer. Stereoisomers have identical bonding relationships, but different spatial positioning. So if you look up here, you've got D-glucose and L-glucose. There's going to be a shift in where those hydroxyl and hydrogen are located. See, they're mirrors of each other. In this case, you have pivots on a central bond. So the bonding relationships are the same, but the spatial positioning, where they're located, is different. All of the bonds, you still got hydrogen attached to carbon, attached to oxygen, attached to hydrogen, but the parts have been shifted a little, moved. Think about these as I think about transformers. Anybody played with transformers when they were little, or like currently? Nobody. Wow. I'm seriously impressed with your lack of childhood. All right, in case you didn't know, because you live under a rock, do you guys know what transformers are? Okay. All right, so you saw the Michael Bay movie, I understand. The deal is, with transformers what happens is each piece pivots. Each piece moves. But it doesn't detach. It doesn't break off and come to a new place. I know in the Michael Bay movie, he totally screwed that up with giving Optimus Prime the ability to fly. Totally terrible scene. But aside from that, all that they did was twist and turn. By twisting and turning, they were able to convert from a robot to a plane. There was no breaking off, coming back together, ripping parts, moving components. It was all just shifting and bending and folding. It's almost like origami in that way. You could take a piece of paper and fold it one way, and you get a crane. You fold it a different way, you get a paper airplane. You fold it a third way, you get a frog. It's the same basic structure. All you're doing is manipulating the spatial positions. An old student of mine, Laura Gibson, except now her name's not Laura Gibson. Now her name is like Laura Haley or something. Anyway, she came up with this idea, and she said, it's cool that you use Starscream and all, but you really should use Bumblebee. Because if you guys know Transformers, what does Bumblebee talk out of? Radio. What's another word for radio? It's stereo, right? Stereoisomer, yeah. So I can't claim credit for that one. I totally give it to one of my students, but I think she was onto something. So there we go. Those are the three important properties of carbon. It forms 4 covalent bonds. It can bond to different functional groups. And it can, carbon-containing molecules can have multiple forms in the form of isomers. You can have stereoisomers, or you can have structural isomers. Do you guys have any questions up till right now? Okay. So we're going to build these molecules. You can sometimes form large molecules by putting lots of small molecules together. This is a picture of a protest that happened on University of Virginia's campus. The president of the campus was ousted. The board of directors, or I forget, board of governors is what they call them, did not like the president, so pretty much they fired her. Well, a student didn't like that. They liked the president. They thought the president was doing a good job. So what they do, they went out and they went to the board of governors and they said, I think your decision is wrong. What did the board of governors do then? You guys have been in this case before, I assume. You, what? Yeah, they said, thank you for your thoughts. We really appreciate your input. Thanks for engaging us in this dialogue. But ultimately didn't care. So that student went and they got more students and more and more. You have a bunch of students and suddenly you've got political power. One student can't do much, but a lot of students is a student body and they can do something. So they protested and eventually the Board of Governors said, oops, we made a mistake. We meant to keep her. And oh, by the way, we happened to get rid of these two guys that were already on the Board of Governors because they were just bad people. Molecules do the same thing. You could take individual small molecules and combine them together to form macromolecules. Macromolecules are just big molecules. They can be made up of monomers. Mono means one. So mono, I assume mer means molecule. Monomer, so one mer, that's what it means. Monomers are small molecules that will link together to form macromolecules. And when you link these monomers together, you form polymers. So up here, the monomer is white, white, black. The polymer is white, white, black, white, white, black, white, white, black, white, white, black. And you see a repeating chain over and over and over again. Monomers come together to form polymers. You guys just aren't getting my pop references. And this one's going to really date me. But monomers and polymers. Do you guys know what this is by chance? Any of you? Yeah, didn't think so. That's what everyone says. It's a Power Ranger. It's not a Power Ranger. This is Voltron, Defender of the Universe. 1984 version. It's totally on Netflix again if you're interested. But what the big thing about this is you'll notice you got, what does this look like right here? What? A dog. Sure, why not? It's a lion, and it's awesome. This one, same thing. Another lion. Another lion, another lion. Even the top's lion. These are 5 monomers that came together to form a polymer. Okay, how about this? So you guys ever seen Power Rangers? How about that? Okay, how many of you guys raise your hand? It's okay, you're allowed. Good. All right, if you've ever seen Power Rangers, all of these Zords, they come together. They always start off individually, right? Which is the stupidest thing in the world because they're never going to beat them all by themselves as single things. And then they all come together to form the Megazord. So you've got individual monomers that can't do much on their own. They come together to form a polymer. Questions on that? Hey, did you guys ever see Power Ranger Time Force? Was that your guys' time? You guys, do you remember? I don't know. Anyways, a friend of mine from high school, turns out she went on to be Jen in that show. So I knew the Pink Power Ranger. A friend of mine got to almost date her, and then he was an idiot and didn't. And I remind him of that every single time I see him. 20 years ago, you remember that time you could have dated the Pink Power Ranger and you didn't? Yeah. How's your life now? He's working at a bookstore. I'm going to have to remember to publish this lecture on YouTube. Send him a link. Just so you know, I'm mocking you in front. They have no idea who you are, but I'm still mocking you. Or I'll send it to his mom. That's even better. So, building a better molecule. I don't know how I got off on that tangent. I'm sorry. Building a better molecule. We need to connect individual monomers together. How are we going to take monomer A and monomer B and put them together? What chemical reaction do you know that can unite two molecules? What? Hydrogen bonds? Okay, yeah, okay, technically. Yeah, thanks for looking at the screen now. Condensation reactions, the big bold term at the top of the slide. It's okay, nobody else noticed it either. That's right, condensation reactions can take two monomers and unite them together to form a polymer. Here we've got one peptide and another peptide, one amino acid, a second amino acid. You'll notice on this side you have a hydroxyl unit hanging off the edge, OH. Over here you have a hydrogen. What's H plus OH going to equal? Water. Thank you. So you've got a bond is going to form between these two peptides, forming a single bond, and water's released. Condensation reaction. Now you've gone from having two small molecules to one larger molecule. We can store energy in bonds. We talked a bit earlier about how rubber bands as bonds, right? And when a rubber band breaks, what happens? You release energy. Somebody gets snapped. When you create macromolecules, you're investing energy to create a bond. And you can use that to store energy. They're like little energy batteries in some cases. It requires energy to build a bond. That bond, that energy stays within the bond until you break it. All right, jumping ahead, how would you break a bond? What would you use to break a bond? Water. And that, what, go ahead. No, not hydrogen. Water. Water breaks a bond. And when water breaks a bond, what's that process? Hydrolysis. So hydrolysis is going to release energy. Condensation reactions, dehydration reactions require energy. Hydrolysis reactions release it, which was right here. Hydrolysis reactions use water as a reactant to split the molecule into its component parts. Here we've got some sucrose, table sugar. It kind of goes around in the plant. It's transported in this way, and then we need to break it down into glucose and fructose. To do that, we add some water, which cuts that bond. When that bond is cut, you release some energy. So hydrolysis releases the energy stored in the bonds that are severed. Condensation reactions store energy, hydrolysis release energy. And I'm harping on that a lot because it comes up again and again and again and again. So we're going to go into the 4 classifications of macromolecules. First off, we've got carbohydrates. You've probably heard of people are like, they talk about them as if they were carbs. Have you heard that? Like, I'm going to cut back on my carbs. I'm going to really eat some carbs today. What they mean is carbohydrates. Carbohydrates are molecules that are composed of carbon, hydrogen, and oxygen, and usually in this ratio of carbon with some number and hydrogen and oxygen as water in some number. So carbon, hydrogen, and oxygen come together to make carbohydrates. Carbon and hydrate water. The monomer of the carbohydrate is the monosaccharide. These are small carbohydrates that taste sweet, and they act as monomers to form larger structures. I don't recommend figuring out whether something is a carbohydrate or not by going and trying to lick it. When in doubt, if you're in chemistry, don't lick it. That's just good advice. If you have already licked it, don't do it again. So you've got small carbohydrates, the monosaccharides coming together to form polysaccharides. You have 2 types of monosaccharides, the pentose sugars and the hexose sugars. Pentose sugars have 5 carbon. Hexose have 6. Think pentose, pentagon, pentagram, something or other. Over here, ribose and deoxyribose. Ribose and deoxyribose are examples of a pentose sugar. Examples of a hexose sugar would be like glucose. So when you think pentose sugar, think 5 carbons. When you think hexose sugar, think 6 carbons. Now, sugar is important. You guys know sugar is important. My God, you're college students. You have to know sugar is important. The big reason sugar is so important is because glucose, which is a very simple sugar and one that we're going to deal with a lot in depth, is small and water soluble. It's got it will allow for water to form around it. That lets it get into the cell very easily. And then there are specialized molecules in the cell that break it down. So the first thing that happens is you're going to have glucose get cut in half. When glucose is cut, what's released? Energy. So you cut this glucose in half. You go from a 6-carbon molecule to a 3-carbon molecule, pyruvate. Then you get this enzyme that's called pyruvate decarboxylase. And it comes along. And it cuts that carbon dioxide off the top. And what's released? Not trying to trick you. You cut a bond. Energy. Whenever you release a bond, whenever you cut a bond, energy is released. Then you're going to hack off a functional group. And what's released? Energy. Do it again and again. And each time that happens, this glucose gives up more of its energy. So it's like a little battery that you can use to power your cells. If you were to have 2 monosaccharides, glucose and glucose, for instance, you can have a dehydration reaction occur to form a disaccharide and eventually a polysaccharide. So here you have 2 molecules, a condensation synthesis reaction that occurs, or a dehydration synthesis reaction occurs to create maltose. Is energy released or stored here? When you're taking 2 molecules and combining them together, do you store energy? A very, very common disaccharide is sucrose. Sucrose is table sugar. It's the stuff that you put on whatever you want to eat, really. This is a major transporter of glucose, and then there's a blank there, but it's through plants. Plants use glucose to move plants use glucose to move sugar around their structures. Basically, in plants, where's sugar made? I'm just referring back to like your elementary school stuff now, so I assume you knew. Okay, how about this? How do plants get their food? Oh, come on, guys. Where do plants get that? What? Okay, sure, from the sun. Let's play this fun game of let's remember elementary school. Yes! Thank you! Photosynthesis, that's all I was looking for. Plants get their food from the Plants get their food from the sun. Sunlight shines down on the plant. Here's our plant. It's happy. It's got some leaf, some roots, dirt. Good so far? Remember this? Flower. We'll have a flower on it too. Flower. Sunlight comes down, strikes the plant. Where does that sunlight get converted into food? And now you're getting really technical. Yes, the chloroplasts. Where are the chloroplasts? I think you said it, but the chloroplasts are in the cells. The cells are where? The stem is kind of green, but not much happens there. In the leaves, right? Sun comes down, strikes the leaves. That's why leaves are big, broad, and flat, so they can get more sunlight. You got this sunlight striking these. They start making sugar. And then the plant doesn't just keep the sugar there. It needs this sugar in the leaves to help all the rest of it grow. So it needs to transport it. To transport that glucose that it just made, it creates sucrose, this water-soluble molecule that gets transported out of the leaf and maybe up to what are called sugar sinks. Might go up to the flower. Maybe that flower starts to grow fruit. And we have, there, big fruit. Might grow, if this is potatoes. You guys know how potato plants work? Oh, we're going to talk about potato plants then. Because now we're just getting way off track. But you need to have sucrose to transport the sugar from one part of the plant to another. You can use that sucrose then to create polysaccharides. If you take lots and lots and lots of monosaccharides and you string them all together, you get a polysaccharide. Here you have glucose, and then it's attached to glucose. You got a covalent bond formed from dehydration synthesis, and then another dehydration synthesis, and another. Each time you're doing this, what are you storing? Energy. So you could have these big, 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 big monosaccharides. Monosaccharides can start forming all sorts of patterns. One is an unbranched pattern. These polysaccharides are going to stack on top of each other. They're almost like corrugated cardboard. So you have a polysaccharide like that, and it's going to come back through and it's going to stack right on top of itself. And it'll stack right on top of itself and keep stacking. That makes it extremely stable. This is used for structure. Cellulose, glycosaminoglycans, chitin. All of these are used for structure. Cellulose is what's used in plants to give it their rigidity. Excuse me. Chitin. Chitin is used in fungus, and another type of chitin is used in insects. Have you ever stepped on a cockroach? Yes, of course you have, because cockroaches are terrible and you want them dead. Okay, how about this? If not a cockroach, at least you've killed a wasp. Or you've watched somebody else do it, because nobody likes wasps. You step on it, and what sound does it make? Crunch. It was able to withstand a lot of your weight because of these overlapping carbohydrates. Another type of branching pattern would be moderately branched. Now, these moderately branched ones look sort of like the unbranched, except they're going to, at points, extend out at angles. So what you'll have is something like this, and then it comes down, and then it goes again like that. So you end up having some branches to it, like a tree branches. Now, the thing is, wherever there's a branch, what can get in there? Water. And what would happen if water got into those branches? It would split it, releasing some energy. Typically, these moderately branched carbohydrates are used for long-term energy storage in plants. Starch. Which gets us back to the potato that you guys don't know about. So, how do potatoes store their energy? Okay, what do you know about potatoes? They make French fries. All right, say it again. They got lots of starch in them. You always hear about that. I mean, these potatoes are starchy. Or, I've eaten a lot of starches. People talk about, they go out to dinner and they're like, I can't eat that potato. I've had too many starches this week. It's like you've had too many carbs. So what happens is, we have our potato. In case you guys didn't know, where do potatoes grow? Underground. But where does the plant grow of the potato? Above the ground, because the potato plant has lots of leaves. What are those leaves doing? Photosynthesis. They're making a lot of sugar. And what they do then is they will send the sugar down to the roots where the potatoes are growing, the tubers. And they start to create big repositories. Now, you're not just going to store glucose by itself. It's going to disappear very quickly. So instead, you convert it to a long-term energy storage solution, starch. You start to build these moderately branched carbohydrates. And as that happens, the potato gets filled up with carbohydrates. Winter comes along. What happens to the plant on top of the ground? That's right. Thanks, Bailey. Appreciate that. Yes, flop. It dies. But what's underground right now? Potatoes. So what happens in spring when it thaws out? You have all this energy stored in here, and the potato starts to put out roots, and it starts to send up a shoot, and life begins again. That's why when you don't eat potatoes and you just let them sit in your house, they start to grow roots. They start to get hairy. They start to prepare to produce a whole new potato. You guys ever seen, was it The Martian? If you haven't, another good movie. He survived because he took potatoes, cut them into smaller potatoes, and was able to plant them and create a whole crop of potatoes. All because of this long-term energy solution in starch. Animals, though, we don't have that. We're going to use this highly branched structure. It's called glycogen. It's this really, really highly branched. Lots of water can get in there and break this apart very easily. Break it down. This is used for energy storage in animals. Long-term energy storage in animals is going to be in fat. We'll get into that soon. Short-term energy storage is glycogen. Now, we're animals. We don't need to store energy for the winter. We try not to anyway. We need it right away because we go and we eat plants. Plants have sugar. We take that sugar, we convert it to glycogen. Then we use that glycogen as fast as we can. It's sort of like the story, do you guys know the story of the grasshopper and the ant and the grasshopper? And the grasshopper played all summer, and the ant work to store up for the winter. In this case, the ant would be the plant. It's storing its energy for the winter, preparing. We are the grasshoppers, playing and frolicking all summer long, eating when we want to, but we're not really storing anything away. And then comes the winter. And what happened? In the story, the grasshopper was starving, and the ants invite him into his house, and the grasshopper learned an important lesson. For us, we just eat the plants. Yeah, they're storing it all winter long. We will dig them up, and then we will eat them because we're the top of the food chain. So this glycogen is used for long-term energy for short-term energy storage in animals. You've got unbranched for structure, moderately branched for long-term energy storage in plants, and highly branched for short-term energy storage in animals. Next up, we've got our lipids. Fats, phospholipids, steroids, and waxes. 4 classifications. I do expect you to know all of them. First off, we have our fats. Fats are known as triglycerides. They contain a molecule of glycerol bound to what are termed 3 fatty acid chains. Now, you guys have seen fats before. What happens when you subject fat to water? Is it hydrophobic, hydrophilic, or amphipathic? It's hydrophobic. Fat does not mix well with water. It does not have polar parts of it. It's made up of a carbon and hydrogen, as you can see here. Carbon, hydrogen, carbon, hydrogen, carbon, hydrogen. Those are similar electronegativity, so they're going to be nonpolar. We have 2 big kinds of fats, saturated fats and unsaturated fats, and I know you've heard of these. Saturated fats. All of them have single bonds straight across. It's a linear molecule. It's straight. In a saturated fat, you're going to have the molecular interaction stabilizing the fat. So it becomes a solid at room temperature. So if you have your triglycerol molecule like that, another triglycerol can link into it. So they're sort of right on top of each other. And you have lots and lots of these reinforcing molecular interactions. So these fats are solid at room temperature. What happens beyond room temperature when it heats up? They melt. Have you ever cooked bacon? If you haven't, I recommend it. It's great smell, great taste. I'm going to have bacon tonight. Bacon wrapped asparagus with a cream cheese center. That's what I'm looking forward to going home. Doesn't it? So what you do with bacon, though, is you cook it. You take this pound of bacon and you throw it into a frying pan, and what happens? The fat melts. And what happens to the bacon? It gets smaller. And don't you feel gypped? Yeah. You're like, I swear there's a pound of this stuff here, and now there's nothing, and I have a whole bunch of this grease, this fat left over. You take the bacon out, and you still have this glob of grease at the bottom. You pull that off the stove, you let it cool down, it solidifies. The molecular interactions reinforce themselves, and you get a solid at room temperature again. Butter, same thing. If you take butter and melt it, you leave it alone for a while, it's going to reform. It doesn't look like butter anymore. But it does reform. It's because all of those bonds reassert themselves and stabilize. So the way I remember saturated fats is saturated fats have single bonds and are solid. Just that easy. All start with the letter S. Saturated, single, solid. Yeah, there we go. Unsaturated fats are going to have a double bond somewhere, and that double bond bends the structure. So an unsaturated fat bends. And when it bends, it means that these fatty acid tails can't connect as well. You have a bend in it, which means that the ones that would normally bind underneath cannot fit in between the spaces, so you don't get them lining up as well. So they are not solid at room temperature. They remain liquids. They keep bouncing off each other. When you think unsaturated fats, think of your Mediterranean dinner table. Nuts, olive oil, things like that. If you've ever eaten a walnut, you open it up, you take a bite, it's wet inside. It's wet not because of water, it's wet because it has unsaturated fats. The way I remember unsaturated fats is it's got a bend to it. Sort of got this U bend, so I think U unsaturated. So there we go, saturated versus unsaturated fats. These saturated fats are used for long-term energy storage in animals. You guys may have noticed over the years that there are probably areas on your body where you're like, huh, that's heavier now than it used to be. That's because that's where you're storing energy for later. Your body is dead set that winter is coming, and you will be prepared. So you store some energy. You set it aside as a fat. Another type of fat are your phospholipids. I'm sorry, another type of lipid is the phospholipid. We talked about phospholipids before. Phospholipids have two nonpolar tails and a phosphate polar head. So you've got a nonpolar side and a polar side. Now, if they're nonpolar and polar at the same time, what does that make them when they deal with water? Amphipathic. In an aqueous environment, these will form either micelles or bilayers. A bilayer is simply like this three-dimensional sphere. You've got water on the inside with your phospholipids facing outwards. I'm sorry, facing inwards, the heads. Then you've got tails, then you've got heads facing out toward water on the outside. You formed a little balloon there. This is how we make cells. Another type of lipid is the steroid. Now, steroids are really useful in animals. I'm not really an animal biologist. I don't do anatomy and physiology. So as far as I'm concerned with you guys, you can think of steroids as they have ring structures. They're not soluble in water, but they're really, really good for intracellular communication. They send messages from one part of your body to another to say turn on or turn off. Estrogen, cholesterol, testosterone, all of these are different types of steroids. So think of them as almost like little text messengers in the cell. They send bits of information from one point of your body to another. And the final class of lipid is the wax. Waxes are nonpolar. All right, nonpolar. How are they going to interact with water? They will not dissolve. So what are they termed? Hydrophobic. Good. These are secreted to prevent water loss. They either keep water on the inside of the plant or sometimes they keep its water out. In plants, when it rains, like it is now, you'll notice on the leaves that there are going to be water droplets on the outside of the leaf. These waxes are preventing the water from getting in. But more importantly, on hot days, those waxes keep water from getting out. It's like having it's sealed. It's almost like a little Ziploc bag for a plant. It keeps water on the inside. So if water was able to escape too quickly, what happens to the plant? It wilts, it dies. Bees. Bees make beeswax. Why do they make beeswax? Everyone's like, to make honey. No. They do it to protect baby bees. Baby bees live in these little combs. And if they were exposed, if they did not live in the combs, these hydrophobic combs, they dry out. They're wet and they're wormy and they don't want to dry. So instead, they need to grow in these little areas that are protected from the elements. All right, so we have two types of macromolecules left to go into: nucleic acids and proteins. Nucleic acids are molecules that are ultimately responsible for storing, transmitting, and expressing genetic information. These are the things that there's a plan to build you. These hold that plan. These are the sort of blueprint to build you. Of the nucleic acids, both are going to be composed of monomers. So you got monomers and polymers. The monomer is that single subunit. The monomer of the nucleotide of the nucleic acid is the nucleotide. So nucleic acids are polymers composed of a whole bunch of nucleotides. If we were to look at just the monomer, we've got, here's a nucleotide, and it's got 3 parts. What do you see as dead center right there in the middle? No, that's over there. Next to the P, over here. Next to the O. There's the P, then the, okay, there's an O, a P, a O, and then a C, right? CH2, that central carbon atom. It's all about carbon-containing molecules. And that carbon is acting like glue, holding this phosphate to that sugar, and attached to that sugar is a nitrogenous base. What kind of sugar is this that has 5 sides? Pentose sugar. So you've got a functional group over here, phosphate, a nitrogen, a sugar attached to it, the carbon, and then a nitrogenous base. That means that it's filled with nitrogen. Those nucleotides are going to covalently bond to each other to form one of 2 kinds of nucleic acids: deoxyribonucleic acid or ribonucleic acid. Deoxyribonucleic acid can also be called DNA. Ribonucleic acid is RNA. Typically, we're not going to say deoxyribonucleic acid. Although, I will say, I totally dominated that Scrabble once because I had a triple word score with nucleic, and then next turn I put ribonucleic, and then I put deoxyribonucleic. I totally cleaned house on that one. The one time I won at Scrabble. So deoxyribonucleic and ribonucleic acid are going to be your 2 types of nucleotides. I'm sorry, acids. Concentrating on DNA to begin with, they're going to be one of 4 structures to fill that nitrogenous base position. All DNA is going to have phosphate, that phosphate functional group, attached to a carbon. Then that carbon will always be attached to deoxyribose, a pentose sugar. But what's attached to that sugar could be either the single-ring thymine, double-ringed adenine, single-ringed cytosine, or double-ringed guanine. We are going to get a lot more into these bases later in the semester, much later in the semester. I just wanted to introduce it now. I guarantee you later on you're going to be seeing these exact same slides because I'm lazy and that's the way I roll when I write my lectures. So this is just to give you a taste of what's to come. 4 structures to create 4 distinct nucleotides in DNA. Those nucleotides then will be able to bond to each other. Adenine binds to thymine, guanosine binds to cytosine. And they're going to bind to each other using hydrogen bonds. Now, individually, are hydrogen bonds strong or weak compared to covalent bonds? Hydrogen bonds are very weak compared to covalent bonds. Covalent bonds are sharing electrons. Hydrogen bonds are a weak interaction between 2 molecules. So here we've got hydrogen bonds holding these 2 strands together. If there's just one hydrogen bond, you can break it pretty easily. Happens all the time with water when it evaporates. But when you have millions and millions and millions of them, suddenly it becomes super strong. It's almost like, you ever had a kid try and hold your hand? Pretty easy to get out of that because you're bigger and stronger than the kid. Just rip your hand away. It's like, don't touch me. But if you've got lots of kids and they're all hanging on to you, much harder to get them off. They are overpowering. You have to like scrape them against the side of something to get them off of you. Think about a caterpillar. Caterpillars have lots and lots and lots of little feet. They can hold on to things very tightly as long as they have multiple feet attaching. But if they only have 1 or 2 attaching, it's very weak. Their caterpillars are pretty weak. So if you have a few hydrogen bonds, it's very weak. But if you have millions, that's a strong connection that stabilizes this double-stranded molecule. Otherwise, there are a bunch of differences between DNA and RNA. The biggest difference that you can see is that DNA is double-stranded versus RNA that's single-stranded. DNA has deoxyribose as the sugar. RNA has ribose. Both are pentose sugars. DNA has thymine as one of its bases. RNA doesn't have thymine. RNA has a totally different base called uracil that looks chemically similar to thymine. And then probably the most important distinction at this level for you guys is DNA stores genetic information, whereas RNA helps to decode the information. DNA is the master plan. RNA is sort of a messenger that delivers the plan to the contractors who can carry it out. This is, of course, for the inevitable question of what's the difference between DNA and RNA. Questions or concerns? All right. Final type of macromolecule. And you will need to know all of these macromolecules, not just for your exam, but your lab next week is all about macromolecules. You're going to be expected to know everything about them going in so that you can manipulate them and do some experiments and figure out what's what. The final classification is proteins. Now, proteins mean literally first rank. They are crucial to survival. They're made up of carbon, nitrogen, oxygen, and hydrogen. It's like most of these organic molecules. Without proteins, you would not be able to survive. Proteins are vital to life. As a rough overview, proteins are made up of amino acids. They have what's termed a hierarchy of structure, and my cop-out answer is that structure is determined by chemistry and physics, because that's biology for you. It's just push it off to the other sciences. So first off, the monomer of the protein is the amino acid. That's otherwise known as a peptide. These amino acids or peptides, whatever you want to call them, are built around, again, a central carbon atom. On one side, you have the amino. On the other side, you have the carboxyl. Where have you seen amino and carboxyl before? The hint is you've seen it at the same place you saw phosphate and methyl and sulfhydryl. Yeah, the functional groups, that's right. You know it, it's okay. There's the amino functional group, the carboxyl functional group. They're binding to a centralized carbon atom. That carbon atom can bind in 4 places. So it's also going to bind up to a hydrogen and to what's known as a variant R side group. There is no element R, by the way. That variant R side group could be lots and lots of different chemical structures. And it's what differentiates different amino acids from each other. There are a grand total of 20 different types of amino acids. I'm going to be really nice and not make you memorize them. But if you look at the very top, you got alanine, valine, leucine, isoleucine, phenylalanine, tryptophan, methionine, proline. All of those are nonpolar. If they're nonpolar, what else does that tell you about them? They don't dissolve in water. They're hydrophobic. They all have those same chemical properties to them. At the top, you see that OOC. That's the carboxyl group attached to a carbon, attached to the amino group. Every single one has that same basic structure. The difference in structure comes on the right side here. Here you got CH3 down here, different structures as you go. Those different structures allow these amino acids to interact with each other in different ways to create unique forms. And if they have unique forms, what else do they have? Unique functions, because form determines function. I'm pretty well going to guarantee you that's going to be on the exam. Form determines function. It should be a dead giveaway answer. As we come down, we've got some of these down here. These are polar and acidic. They give off hydrogen ions in solution. How will they interact with water if they're polar? They dissolve readily. They're hydrophilic. Up here we have polar and uncharged, and then polar basic. Again, these are all hydrophilic. The polar basic ones give off or will absorb hydrogen ions. So 20 different types of amino acids, all with different ways of interacting with each other. You can take any two of these amino acids and combine them together. You're going to use dehydration reactions or condensation synthesis. So if you've got this amino acid here and an amino acid here, they come together, water's released, and you formed a peptide bond, otherwise known as a covalent bond. I have a friend who's he's a metallo-organic chemist, and I asked him one time, what's the difference between a peptide bond and a covalent bond? And he said, well, one forms between peptides and the other doesn't. And I said, no, but seriously, what's the difference? And he said, well, there is none. And I was like, so it's just a really pretentious way of saying covalent bond. This is just a regular old covalent bond. They're just sharing electrons linked together. You can begin linking peptide after peptide after peptide, amino acid after amino acid. Between these two, the water's removed, covalent bond is formed. Water's removed, covalent bond is formed. Water is removed, covalent bond is formed. It's all about dehydration synthesis or condensation synthesis, dehydration reactions. Does this release or store energy when you do this? Why? Because they're coming together. And anytime you form a bond, you need energy. You release energy when you break a bond. What kind of reaction would release energy? Hydrolysis reactions. All right, so now we've got these amino acids all lined up. Individual peptides are the monomer to form this longer structure, which is going to be the polymer known as a polypeptide. Lots of peptides together make a polypeptide. Each of them has these variant R side chains that are slightly different. The interaction between these R side chains is what gives a protein its shape. Because right now we have a straight line polypeptide. It's got to fold up and associate with itself in such a way that it makes this unique structure. So polypeptides go on to become proteins. The polypeptide is just a big, long chain of amino acids. The protein is a long chain of amino acids that's folded into a three-dimensional shape. So polypeptides are like the first level of organization. The final level will be the protein. These proteins, the final folded up form of the polypeptide, is going to be used in a lot of different things. And I will challenge you, but I think, as far as I know, every reaction in your body, every reaction dealing with life is somehow mediated by a protein. Proteins are involved in just about everything. Proteins are involved in expressing your genes. They help build DNA. They build RNA. They build other proteins. They regulate. They turn on and turn off genes. Here's this double-stranded nucleic acid. What is it? DNA. And you'll notice that DNA has these sort of major and minor grooves in it. Those grooves are an open spot where a protein can bind. Form determines function there. They help move things around the cell. They initiate movement. Proteins are used. This is a dynein protein and kinesin. This is used to walk things around the cell. Literally, they walk like this sort of motion like that. It's like kinase on, kinase off, kinase on, kinase off. And they walk along these things called microtubules we'll learn about shortly. But you can have movement because of proteins. Protect yourself from disease. Oftentimes, viruses will come and insert their own foreign DNA into your body. And that DNA will start to replicate itself and create viral proteins. Your body has its own proteins to fight back. Sort of like what they do is they'll discover that viral DNA and then chop it into little pieces. It's with a a protein called dicer. It's sort of like the fruit ninja of proteins. You can have enzymes that increase the rate of chemical reactions, that help cells talk back and forth with each other, and that give support and strength to structures. Chromosomes, that genetic material in the center of the cell, are made up of very, very tightly coiled up DNA. You wouldn't be able to tightly coil it up without proteins providing the scaffolding. You have things to help chemicals in and out of the cell. Proteins will open doors. They let some things in, they don't let other things in. Proteins act as conveyor belts to move things from one part of the cell to another. They transport materials around. Ultimately, what it's going to give any protein its function of anything from that list is going to be its form. And that form is going to be determined by what's known as the hierarchy of structure. You've got a primary structure, secondary, tertiary, and quaternary. For those of you keeping track, this will be an essay question on your exam. The question will be, and I'm flat out telling you, the question will be, describe the hierarchy of structure in proteins. So first off, we have the primary structure. The primary structure of the protein is just the amino acid sequence, just the polypeptide. That's it. What amino acid is attached to what other amino acid, forming a big, long, straight line. So you've got a primary sequence. It's a straight line. So far, so good. Secondary structures begin to form when the amino acids interact with each other. So let's say at one point in this line we've got a positive charge, one point we've got a negative charge. What are these two going to do to each other? They'll attract each other, which might make a loop. Does that make sense? That amino acid and that amino acid are interacting. You might form a structure called an alpha helix. An alpha helix is a big, long squiggle. The amino acid sequence forces the polypeptide to twist itself into a helix. It's almost like a slinky. You might form beta sheets. In this case, the amino acids are going to interact in such a way as to create these zigzag shapes. So you'll either have secondary structures of alpha helixes or beta sheets. It is just the amino acids interacting with each other. No, it is just a helical shape. Next up, you have your tertiary structure. Your tertiary structure is where your secondary structures interact with each other. So whereas before you had alpha helixes and beta sheets separate, now they start to interact to form a tertiary structure. Now you have a three-dimensional shape. Some proteins stop there. Others go on to form a quaternary structure. Oh, sorry, yes? Just the same polypeptide interacting with itself. The quaternary structure is going to be two polypeptides coming together to form a single functional unit. So now you've got a tertiary structure and another tertiary structure interacting to form a complex three-dimensional shape. So primary structure, big long line of amino acids. Secondary structure are the amino acids interacting. Tertiary structures are formed when the secondary structures interact, and quaternary structures are formed when the tertiary structures interact. Then you have a complex three-dimensional shape that has a form and a function. All right, so I got this phone cord. I got it off my office phone. Perhaps you guys have seen phone cords before. Back in the days of the dinosaurs, phones had tails. They were attached to the wall. I don't know if you guys remember that or not. So if you had this phone cord attached to the wall, it's got this bit at the end that's all coily. The phone cord itself is actually one strong, straight, long line. Does that make sense? This would be its primary structure. There's wire in there that's going to interact with itself. When it starts to interact with itself, you get these coils. This is the secondary structure. Tertiary structures form when this interacts with itself. Now, if you've ever talked on the phone with one of these at some point in your life, you know that it starts to twist itself up. The only way to untwist it is sort of just to let it hang. Questions so far? That's tertiary structure. A quaternary structure would be if I had two of these cords and they wrapped around each other. It creates a complex three-dimensional shape. So primary, straight line. Secondary, coils up. Tertiary, it interacts with itself. Quaternary, two or more of these interact. So that is the hierarchy of structure. Any questions? We've got one more super fun example. So this is a balloon animal kit I got from a student. Fun story. So at DCC, we have these things called APDUs, and these are performance goals that you're supposed to meet every year. Goals that you have to have in order to continue working at the college. You have to meet them, right? Well, I didn't think anybody ever read them. So I wrote down I was going to learn how to make balloon animals as my professional goal for the year. Turns out, Dr. Fox, he's the British guy who lives down the hall, he actually reads those things. So he called me into his office and said, Professor Pantasis, you wrote down that you were going to make balloon animals, 3 different kinds. Show me that you can make 3 different balloon animals. So I quickly went back to my office and learned. He said, now that you've shown me how you can make balloon animals, explain, how does this relate to biology? So, because I ended up putting it on this APDU and got called out on it, I'll teach you guys what I figured out. This is a polypeptide sequence, one long chain of amino acids, right? Those amino acids begin to interact with each other. To form secondary structures. These are just the amino acids interacting. Those amino acids interacting, those secondary structures come together to form tertiary structures. These formed from the secondary structures interacting. And you just keep going, because these proteins are going to be much more complex than that. To form a tertiary structure. Now, a quaternary structure is going to be 2 or more tertiary structures working together. So if you've got one tertiary structure and a second tertiary structure, they interact. To form a quaternary structure. Does that make sense? Here you go. No problem. All right, so primary, secondary, tertiary, quaternary. Those are the hierarchy of structure of proteins. These proteins, the structure, these connections between amino acids are going to be determined by one of, or actually multiple, different methods. Good news is, you know most of these already. Proteins can be united using hydrogen bonds, ionic bonds, hydrophobic interactions, which is one we'll get into in a second, van der Waals forces, which some of you guys asked me about earlier in the year, and disulfide bridges, which are really just covalent bonds. So first off, we can have amino acids connecting to each other through hydrogen bonds. Weak hydrogen bonds between amino acids can stabilize secondary structures in proteins. Here you've got oxygen, which is extremely electronegative, and hydrogen, which is less electronegative. you're going to have a very sort of a negative aspect to that oxygen, a positive aspect to that hydrogen, which is going to have different charges and opposite charges attract. So you've stabilized the protein, the shape. You'll notice how they're not right next to each other. They're distant from each other on the polypeptide. These hydrogen bonds tend to stabilize alpha helixes. They're what keep everything bound tightly together. You might have ionic bonds. Ionic bonds work sort of the same way. You've got one amino acid that's charged positive, one that's charged negative. They attract each other. They hold each other tight. You might have what's called the hydrophobic effect. Non-polar amino acids, if they're non-polar, how would they interact with water? They don't dissolve. They're hydrophobic. They're going to exclude water. They're hydrophobic, which causes them that when you drop this into an aqueous solution, all of those non-polar amino acids move to the inside to be protected. All the hydrophilic ones move to the outside. So here you've got all the white amino acids are hydrophilic, all the black ones are hydrophobic. So when you drop it in water, all of the white ones move to the outside, all the black ones move to the inside. The hydrophilic ones face water, the hydrophobic ones face away from it. The way I always think about this is, imagine, if you will, you guys know cats, right? Cats and dogs. What do dogs think of water? They love water. If you put a dog out by a creek, they will come back muddy every single time. They love water. Cats, how about them? They hate water. So imagine, if you will, you took a bunch of cats and dogs and you stapled them nose to tail. It's not an ethical thing to do. Don't do it. But imagine you did. If you took these dogs and cats and stapled them nose to tail and threw them in the water, the dogs would splash playfully in the water. The cats would want to get out of the water. That leads to forming a raft of dogs with cats on top of them. Does that make sense? You've automatically formed a structure by how it interacts with water. The hydrophilic molecules will associate toward the water, the hydrophobic ones away from it. And that's known as the hydrophobic effect. That's another way of stabilizing protein shape. You might have van der Waals forces. Van der Waals forces are extremely weak. They are the weakest of the molecular interactions. Effectively, atoms can attract or repel each other based off of random movement of electrons. Just by chance, there may be more electrons on one side of an atom than another at a given time. And when that happens, it's weakly going to be attracted to a more positively charged atom. Or it'll start to repel the electrons from another atom, creating another temporary dipole. This is an extremely weak force, but when taken into account with billions and billions of atoms, it becomes stronger. Finally, we have disulfide bridges. Here, the amino acid cysteine has sulfhydryl groups. These SH groups, sulfur and hydrogen. Those sulfhydryl groups can covalently bond to each other, forming extremely tight connections between the amino acids. So, 5 forces that affect protein structure: hydrogen bonds, hydrophobic effect, ionic bonds, disulfide bridges, and van der Waals forces. In order of strength, you'll have your covalent bonds as the strongest, your disulfide bridges, your ionic bonds, your hydrophobic effect, your hydrogen bonds, and your van der Waals forces. So now you've got this 3-dimensional protein. All those amino acids have interacted with each other. They form secondary structures, alpha helices and beta sheets. Those have gone on to form tertiary structures. And those tertiary structures interact with each other to form quaternary structures. Now we can have proteins that interact with other proteins. You've got a fully formed protein. It can work with a whole nother protein. The surface of one protein has all of these amino acids that can then interact with the surface of another protein, forming a very complex shape. these proteins working together can then modify biological processes. A lot of what we're going to talk about going forward are going to be proteins interacting with proteins and proteins interacting with individual molecules for something to happen. There we go. Do you guys have any questions?